Hello everyone, my name is Chethan Kumar. Now I am presenting my research findings which shows the evidence for the mechanism of bacteriophage lytic enzyme in phage therapy against streptococcal infection by in silico approach, means by a computational methodology. Nowadays, the bacterial infections, especially in the diabetic type 2 diabetic foot ulcers, foot infections, is a really challenging for all the clinicians who are managing these kind of infections. Why I am mentioning this as a challenging means because of the multi-drug resistant bacteria which are involved in these kind of infections. Coming to my introduction of this presentation, as I said, rising prevalence of antibiotic resistance bacteria became a major public health concern. Yes, as I mentioned, not only in the diabetic foot ulcers, in many other infections, these these antibiotic resistance bacteria become a major problem in public health. So next, the alternatives to treatment with antibiotics are the major research area for the researchers across the globe. Yes, across the globe, many researchers who are working in this domain, they are finding many alternate ways to this kind of treatment for not using the antibiotics. The possible therapeutic use of bacteriophages, that is the phage therapy against pathogenic bacteria to cure infections, are gaining attention. Bacteriophage is the one therapy which is extensively trying in a preclinical model, that's in animal models. In our site also we have tried some experiments on animal models regarding these infections, bacterial infections, and we got a very promising results that we got published in very good international journals also. But we are not aware of the evidence. We are not having the evidence for the probable mode of action is still unclear for us. So for that reason, I choose this area. It can provide the evidence through an in silico approach. Many literatures, many papers will explain about the probable mode of actions, but not the evidence. So when coming to the phage, bacteriophage is a virus which replicates inside the bacteria host cell. So bacteria, we can see in the picture, it's sitting above the bacteria. The bacteriophage typically includes two things, that is holins and lysins, as they are part of their lytic system to achieve virus exist from the host bacterial cell. So holins are responsible for forming pores in the cytoplasmic membrane, and whereas the lysins are responsible for the degradation of the peptidoglycan layer, that is the cell wall of the bacteria. This peptidoglycan is present at the cell wall of the bacteria. That degradation will happen, and the virus, the genetic material, and it gets replicated inside the cell, and it degrades the wall, and thus the bacteria dies. When come to the lysine, when the lysine, the rapid due to the activity of lysine, the rapid cell rupture and the concomitant virus released through the loss of osmotic integrity. So here the lysine plays a very important role in the lysine of bacterial cell wall. That results the potential therapeutic effect against multi-drug resistant bacteria in foot infections, especially type 2 diabetes mellitus. These concepts are used by many researchers as to gain the therapeutic effect as a treatment, as a therapy. They tried to use. As I said earlier, there is no evidence how the mechanism goes on. So my interest was on the bacteriophage lysine, the lysine part. When we go deep into the lysine, lysines are generally composed of a single polypeptide consisting of two domains that we can clearly see in the picture. That is, the one is N terminal and the C terminal. This N terminal is very important as it is a catalytic domain, and the C terminal is the binding domain which attaches to the wall of bacteria. And these catalytic domain N terminal represents peptidoglycan hydrolase activities in the cell wall of the bacteria. So for the present study, I have chosen the Streptococcal C1 phage lysine PLYC. Why it is called as Streptococcal C1 phage lysine is this particular virus will replicate in the Streptococcal bacteria. So that phage is will be called as a Streptococcal C1 phage lysine. So in that lysine, PLYC in a short form, PLYC is unique among the gram-positive lysine. In that it consists of two separate PLCA and PLCB, two separate units. That we can clearly differentiate in the figure. That PLCA is known to contain active cysteine histidine dependent amino hydrolase peptidase. In short, we can call it as a CHAP domain, which is very important for the responsible of amidase catalytic activity. We can notice in the green structure in the figure, which written as CHAP PLYCA and with a particular amino acid number. My study was involved how this mechanism, this domain will help to show this therapeutic effect. As a part of study design, I've used some computational methods I have done the docking studies to see the interactions between these molecules. Docking studies is nothing but a computer simulation based program in that we can understand or we can see the interaction between the two molecules. That is, especially we do it in the drug discovery process. One is as a ligand and a receptor protein. Here, as a protein, I have used as a lysine, which I have retrieved from the protein data bank. It's a universal, free accessible data bank, protein data bank, which was, the experimentally solved X-ray crystallography or NMR data will be submitted by the structural biologist across the world. So I have used this protein data bank to retrieve the structure of three-dimensional structure of lysine, which is a protein produced by the bacteriophage. And as a ligand, I have used the peptidoglycan. Peptidoglycan is here as used as a ligand, but it is present at the cell wall of the bacteria. So the interaction between these two was studied by using the computer simulation technique called the docking studies. And here the peptidoglycan comes with the extension file of SDF that I have converted using one more software called OpenBabel software. It converts both the files into PDB format. That format need to be present for do the docking studies. And the docking studies was I have used the one software called Hex 6.3 software for the to see the interactions, and the results were visualized using a molecular visualization software called PyMol that I have used during this study. As I mentioned earlier, this is the you can see the three-dimensional structure of lysine that we can rotate 360 degree in the software, which is having a unique ID 4F88. We can if we go and type this ID, we will get the structure freely in the database, which was retrieved from the Streptococcus phage C1, which is having the sequence ID of Q7Y3F1. and having a total of amino acids for 465 amino acids are present. Similarly, as a ligand, I have used peptidoglycan as I mentioned in my earlier slide. For this, I have retrieved this three-dimensional structure from the PubChem. It's again a database maintained by NCBI, National Center for Biotechnology Information, under the NLM USA. with having the accession number ID of 9816401. These molecular docking study findings revealed that the oxygen atom of the peptidoglycan is showing a hydrophilic interaction with the hydrogen atom of valine 38 in the lysine protein. This confirms there is an interaction between the ligand and the protein. Again, we can see this image in the next slide. Yes, valine 38 lies, very interestingly, this valine 388 lies in the chap domain.